In this video, we will discuss about how the researchers has improved the iCAR-CTL efficiencies against the tumors. In the previous video, we have seen that this iCAR-T-CTL, that is the T cells derived from iPSCs, or the mature T cells derived from mature T cell lineage derived from iPSCs, then were converted into these CAR-T cells, and those CAR-T cells does not perform well in the in vivo studies. So they decided to improve its efficiency. So first they tried it by inhibiting the PD-1 signalings in the iPSCs. However, that did not work, and those results are in the supplemental figures. Thereafter, they tried it by inhibiting the DAG kinase, that is, it has two different isoforms, DGK alpha and DGK beta. It will so this kinase usually degrade DAG, thereby preventing MEK ERK pathways and the NF kappa beta activations. By inhibiting this DGK alpha and DGK beta, they are not able to degrade DAG, and thereby it will lead to the increased it should lead to the increased MEK ERK phosphorylations. So they use the CRISPR-Cas9 to knock down DGK alpha and DGK beta in the iPSCs, and then did the differentiations into the towards the T cell lineage, and then make the iCAR-TCL from the mature T cell lineage, and then check the ERK phosphorylations, and they are able to see increase in the ERK phosphorylations compared to the iCAR-TCTL that does not have DGK alpha and DGK beta, and those increased is comparable to the CAR-T cells that are made from PBMCs. Thereafter, they wanted to check its efficiencies against the tumors, so they did it using three different conditions. First, they used the iCAR-TCTL that they used in the previous studies, and then they used the modified, in which the DGK kinase is being knocked down, and then we compare it with the CAR-Ts that are formed from PBMCs, and they are able to see the tumors suppressions works well in the DGK knockdown and the primary CAR-T cells from PBMCs, whereas they are able to see the signals for the tumors in the iCAR-T and the control, that is PBS. The quantifications are shown in the panel E, where we can see that the performance of the DGK knockdown CAR-T cells and the pCAR-T cells are similar and better compared to the control case and to the case where they use iCAR-T that has the DGK. And they also checked the proliferations using BRDO or thymidine incorporations, and they are able to see increased proliferations of the DGK knockdown iCAR-T cells compared to the iCAR-TCTL T cells. And when they check the survival rates also shows the similar results that they seen in the tumor total body flux, that survival rates are high for the CAR-T cells from the PBMCs and the CAR-T cells from the induced pluripotent stem cells with DGK knockdown. So once validating its efficiencies, improved efficiencies against the tumors, they wanted to check how long these CAR-T cells are able to survive within the patients who are affected with the tumors. So they did it. They compared the studies with the iCAR-T cells and their actual case in which DGK has been knocked down. And they are able to see that when the DGK has been knocked down, they are able to the amount of CAR-T cells retained in the time of CAR-T cells retained in this conditions is higher compared to the case where normal CAR-T cells from the mature T cell lineage from iPSCs are used. The quantifications are shown in the panel I, where we can see that at day 14 the bioluminescence signals of the T cells are almost similar. At 21 also the difference is not statistically significant, but at 28 the difference becomes statistically significant, indicating that the modifications of inhibiting or knocking down DGK alpha and DGK beta helps in improving the efficiencies of iCAR-TCTL derived from mature T cell lineage from induced pluripotent stem cells. They knock down this DGK alpha and DGK beta in the induced pluripotent stem cells, so that the final T cell lineage does not have these isoforms or this DAG kinase. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.